Okay, so hi everyone. This is the fifth lecture for algorithms describing protein structures. And so today we are going to talk about the complexity that is associated to protein structures and function and interactions. So this is the typical classical paradigm in biology where we assume that a chunk of DNA in the genome, which is called a gene, encodes for a protein which will adopt in solution a particular 3D structure, and that structure will determine a specific function. So here there is an obvious element that is missing, and I think now you should know by now because we talked about this a lot. It's the dynamical aspects of protein structure. So the fact that protein structures are not static objects, but they constantly move, fluctuate in solution, and these motions, these fluctuations are associated to its function. So here I show the example of the famous p53 protein, which is a tumor suppressor, and it has a domain that is what is called intrinsically disordered, meaning it doesn't adopt a particular conformation in solution. So it's literally, it can span all these spaghetti conformations, if you wish, and it will locally fold upon interaction with different partners. And it can interact with hundreds of different partners. So protein disorder is some extreme in the spectrum of motions and fluctuations we can have. So from a very stable 3D folded 3D structure to protein disorder, there is a whole spectrum of different types of motions and fluctuation. So this disorder may concern an entire protein or just a portion of the protein. So you may think of actually two dimensions, two spectra with two dimensions. So you have the spectra between what is ordered and what is disordered, right? With different, what I mentioned before. And then you have another dimension, which is the extent of disorder or the extent of fluctuation that you will have in the protein. So here, that's what you see as the other dimension. You start from a completely structured protein, and then you add some parts that are disordered and that are more or less disordered because they may have local structures, they may fold upon binding, etc. So in the human proteome, it is estimated that disorder is included in more than 50% of human proteins. So that's a lot. We have some disorder in more than half of the proteins. So what kind of things we can have with this disorder? So it can, for example, facilitate the regulation of proteins via diverse post-translational modification. So these disordered regions are typically prone to contain post-translational modification sites, and these sites are implicated in the regulation of the protein. So the fact that they're disordered also may make these sites more exposed to regulators. They may also have a role in the scaffolding and recruitment of different binding partners. So typically you will have these disordered parts that will fold locally to bind some partner, and they also confer to the protein some conformational variability and adaptability. So that's a level of flexibility that the protein has to adapt to different shapes. So again, if we go back to the example of p53, what you have here are examples of protein of complex structures, 3D structures of p53, one domain of p53 in interactions with different partners. Well, not one domain, but different parts of p53. Okay, So on the plot you have the residue numbers, so this is really the sequence index. So 400 residues, and you have the probability of being folded. So the pond R score will give you an indication of whether the structure is disordered, that the protein is expected to be disordered at this region or not. And then the colors refer to the different partners. So you have some parts in the middle, which you can see is folded, right? So the ponder score is mostly negative in this part, in this blue region. And this corresponds to 4 partners. So 4 complexes, 2 on the top, 2 at the bottom, where you can see the structure of P53 domain, which is ordered, which is represented in cyan, cartoon, and then you have the partners in surface. So you can see the partners are very different. Mostly they bind to the same region, right? So it can be a bit more extended, etc., but it's mostly the same region. And then you have more disordered parts where the ponder score is high, so that's at the N-terminal and C-terminal part. And in those parts you also have several partners, so 3 on the left and 4, more than 4, 1, 2, 3, 4, 5, 6, 7, 7 on the right. And in these structures you see just a part of P53 that is ordered, which correspond really to the interaction site with the partner. What is also interesting is on the far right, you can see that you have these colored squares that are on top of each other. So these correspond to different partners binding really to the same position. And when you look at the structure, you can see that this position, this segment has different conformations depending on the partner. So it's not only that it folds upon binding, but it also can fold in different conformation depending on the partner. So that's really, here I think you really see the complexity that you can have, and it's a huge complexity. So there is complexity at this level, which is the most obvious for us for this course, let's say. But there is also complexity at other levels in this paradigm. So what about sequences? In fact, a gene is not just a chunk of DNA that uniquely encodes for one protein. The gene is comprised of blocks, which are the exons, separated by introns, and during the transcription, different sets of exons will potentially lead to different transcripts. So that's what you see here for a protein kinase, which is the C-gene N-terminal kinase. Number 1, it's a human protein. So in eukaryotes, virtually all multi-exon gene will lead to different transcripts. So here you have 1, 2, 3, 4, 5, 6, 7 isoforms, 7 different transcripts that are presented on the left, and they correspond, as you can see, to different collections, different sets of the exons that comprise the gene. And these different transcripts may be translated in different proteins, which may adopt different 3D structures. So on the right you have models, 3D models corresponding to the proteins. So there is one change which is well documented and which correspond to this colored helix. So here you have no change apparently in the structure, but just subtle differences in the side chains of the residues and the exposition of these side chains. Or you may have big changes like these big truncation that you can see at the bottom. And you see it also on the left, you have small versions of the protein, for example. So then the question is, are these functional? Are these really translated? That's another question, but there is evidence for several genes of different versions, different isoforms of the proteins playing different function, different role in the cell, so having different partners, for example. So this is certainly a regulatory mechanism that should be taken into account when we look at the structure and then the function. So what about function now? We may have some strange things like here. So you see 2 proteins which are composed of 2 domains, and the 2 domains are colored in different colors. So on top, this is the acotinase enzyme. It's a key player in the central pathway of energy production. So it binds citrates and converts it into isocitrates. And at the bottom, we have a transcription factor. So it's a protein that interacts with RNA and which will control the levels of iron inside cells. So we have two different partners, right? A small compound, chemical compound on top, and RNA at the bottom. We have different shapes. But if we look closely, what we realize is that these two proteins are in fact the same protein. You just have the top domain in blue, which is closed on top, and then open at the bottom. So it's just one protein performing completely different functions in different contexts. So they were discovered kind of separately, and then people figured out it was the same. So these proteins are called moonlighting proteins, meaning they have their official job and some undercover job. And we have examples of them, but there may be much more than what we know right now. And the change is really big. I mean, we go from an enzyme, which catalyzes a chemical reaction, to just binding to RNA. So, okay, so going a bit further in this complexity idea, we should not consider proteins on their own, because they interact with other proteins all the time, with DNA, with RNA, with chemical compounds, and that's really the essence of cellular function, these interactions. So we can think of it as the society of proteins. What is important is that the cell is a very crowded environment. So here you have a picture, a snapshot showing you some, so it's a study that dates back now 10 years ago, of Brownian dynamic simulations of an E. coli cell. So you have some nucleic acid and also some proteins. What you can see is that it looks like the subway when it's crowded, right? So, and this is realistic in terms of concentration. So it means that proteins encounter each other all the time. And yet, in this crowded environment, they are able to perform specific functions by binding to specific partners. So on the right, you have a representation, a graph representation of interactions between different proteins of an organism. So here it's the yeast interactome. So it's a graph that is determined based on experiments. But in fact, so you have to keep in mind that there are many interactions that can be detected experimentally, but we are far from knowing really all these interactions and from having some structural details about them. And also that there are some discrepancies between different experiments and false positives. Or at least there is a debate about whether these detected interactions are false positives or not. And that experimentally also it's very difficult to determine with certainty that two proteins cannot interact. So this brings us to a more computational point of view, let's say, with the docking problem. So what is this problem? It was formulated now in the 80s, so quite some time ago. Given the structures of any two proteins, the question is it possible to predict whether they associate? And if they associate, in what way they associate? So in 86, Connolly was saying that within a few years more development, there's then a good chance of solving the protein docking problem. So there is the folding problem, which we saw before, and now that's the docking problem. So this problem has motivated the development of docking algorithms. And so this problem of docking molecules of any complexity based on the complementarity of the features has been shown to be complete. So when you see the original formulation, you see that the problem includes whether they associate and how. So docking algorithms have mainly focused on answering the second question, in what way, how. So meaning that a typical docking algorithm will take two proteins as inputs, knowing that they should interact, that they interact in some condition, and the algorithm will try to find the optimum position and orientation of the two proteins with respect to each other. So that's a bit of sub-problem, right? Because asking whether they interact it's a very difficult question. So if we think of molecular docking, let's say we have protein, which we call L, because by convention we will define ligand, and R, the other one, which will be the receptor. So that's really conventional names. They doesn't reflect function, but... And we want to know how they associate. Okay. So the point is you may have to associate to undergo conformational changes. So again, protein 3D structures are not static. They may encounter motions, they may fluctuate, and they may change a lot to be able to associate with each other. So I should show, I think, the complex. So if you start from these guys and you try to put them together, if the truth is with this change, then you will have clashes in your complex. So docking is a difficult problem. So how do we solve it? We need to define a representation for the system, as in the exploration of the conformational space, which we saw previously. And again, similarly to what we saw previously, you need a strategy to search the conformational space and a scoring function to evaluate the conformations you're generating. So here we will talk about the docking step and the scoring step. So the space search is a matter of size. For two rigid proteins, not accounting for conformational changes, docking in itself is a 6-dimensional search problem. But as we said, proteins are flexible, so in fact, if we would consider this flexibility, we completely explode the search space. So in the case of a small molecule binding to a protein, exploring the full conformational space of that small molecule is feasible. But in the case of proteins, a bit more complex. So this is a typical workflow. You have the two molecules. For example, you perform a rigid body search, so consider them as rigid objects. You just have your 6D exploration, generate possible complexes, rerank the complexes based on your scoring function or binding energy that you estimate with the scoring function. You may introduce some flexibility, refine, and re-rank. So let's say you select a subset of candidates on which you apply conformational changes, flexibility to refine, and to get the final answer. And you may include at any step some experimental information, for example, contacts that you know from cross-linking experiments. So there are different paradigms here that have been formulated. The simplest case is the lock and key mechanism. So that's also historically the first one that was proposed. You just think of the two proteins as a lock and a key which will enter into it. So here we are in a rigid body framework paradigm. Now we may think of these conformational changes in the way that the two proteins will rearrange themselves upon binding. Because they bind, then there will be some induced fits. And the third one is also accounting for conformational changes. So here the idea is more that proteins live in a pre-existing equilibrium between different conformations. And that's more like an ensemble view and a statistical view, which is probably more realistic than the other ones. And upon binding, there will be some change in this landscape and some conformation that will be selected that will be favored from an energetical point of view to bind to the partner. Okay, so we may think that the binding process is a mix of these contributions. So it's not very clear. It's a view of the mind, like the view we have about the funneled energy landscape. But this are interesting. You can validate some mechanism by doing some kinetics on the association. Okay, so for example, here you have a scheme where you combine induced fit and conformational selection. So let's say you have your P1 and the ligand. And P1 can adopt two different conformations. So you have first the pre-equilibrium between the two, and you can see that the one in purple is more favored than the one in orange. And then when you associate with the ligand, it's the opposite. So the one in orange is more favored than the one in purple, because the one in purple doesn't really fit with the ligand, right? So you may still have the ligand that binds to the purple one, and then it will produce conformational change. So there is conformational selection and also conformational change upon binding, which is called in use fit. And the kinetic rate constants can dictate which molecular pathway will dominate. Okay, so then we are facing a puzzle. Why? Because, well, we may think of two proteins interacting with each other. We may think of many, many more. So we have here, for example, 1, 2, 3, 4, 5 proteins, and they were supposed to interact. And they move, so we are not really sure of their conformation. They need to encounter some conformational change, so the puzzle is not really okay at first, and then with induced fit for the reorganization, it can be. Okay, so the most obvious way to account for these recognition mechanisms is to try to find the complementarity, whether the surface of the proteins are complementary. So we need to have a description of the molecular surface. So how this can be computed? Well, the idea is that you can take a probe, which is just a ball, a pseudoatom if you wish, and you roll it over the protein. So here the red discs, the red spheres, correspond to the volumes that every protein atom takes. So that's defined by the van der Waals radius. So you can simply roll that probe such that it is always in contact with this excluded volumes. And you can see that the thin blue line will correspond to will define some surface, right? And this is the molecular surface. And then if you add the radius of the probe, you define the solvent accessible surface, which is actually the surface that can be touched by water. Okay, so this volume that is included in the molecular surface is not penetrable by water. So you may also use a description where you consider knobs and holes. So holes are concave parts of the surface and knobs are convex parts. And the criteria for a good surface shape measure for docking are that it should be local, so not independent of distant regions of the protein. It should be independent of the coordinate system, and it should allow you to determine whether the shapes are complementary in a fast way. So here, for example, you have a representation of the protein, which is of the surface, which is triangulated using triangles, right? So you take the surface accessible, the solvent accessible surface, you triangulate it, you obtain a polyhedron, and then you can calculate the shape function at each vertex of the polyhedron and compare, for example, the values that you have with neighboring vertices. So this way you are able to identify critical points, which are exactly the knobs and holes I was talking about, and which correspond to local extrema of the shape function. So locally you will have some protruding part or some convex or concave, and these correspond to the critical points. So here is an example of docking program, which is called DOC, which was developed in the 80s by the Kunz group at UCSF. And originally DOC was using a geometric matching algorithm to superimpose the ligand onto a negative image of the binding pocket. So you start with your protein, you determine some binding pockets that you have to detect, which correspond to cavity. Then you take the negative image of this pocket, that's the blue mesh, and then you are trying to put your ligand, your small molecule, inside this negative image of the binding pocket. Here is another example, which is called HEX, which uses spherical harmonics to represent protein surfaces. So the idea is that you just decompose your surface you're using a basis set to decompose your surface, and these functions in your basis set are spherical harmonics. So you have some examples here of what shape they have. And so you can determine the level of resolution you're going to have on the surface by choosing different numbers by fixing n, right? So the number of spherical harmonics you're going to consider will determine whether you have a well-defined, very detailed envelope or a rough envelope. And so by playing with this level of resolution, you can also play with the efficiency with which you compare your surfaces. So Hex using a double skin shape model. So I don't know if I'm talking about this now. So the idea is that you have an external shape, an external skin and an internal skin, and you're trying to maximize the match between the external skin of one protein and the internal skin of the other one, right? If you have the two internal skin touching each other, this is a steric clash. If it's just the external skin, it's not enough. So you are going to play with the angles of the two partners with respect to each other and the distance you have between the center masses. So typically what Hex does is first scan at low resolution with a few functions in the basis set, and it can generate this with billions of orientations in 1 second, in seconds. And then it will refine with a higher resolution. So if you only know one key residue, you can drastically reduce that search space. So if you know some context, it can be very useful. So then you need to estimate the binding affinity. So as I mentioned, you can use the geometric complementarity of the molecular surfaces, and you can use also other ingredients, like heuristic scores based on the number of residue contacts, like potentials of mean force or statistical potentials that are derived from database knowledge. You can have a physics-based function, very similar to the force fields that we saw, so using terms that represent the van der Waals interactions, the electrostatic interaction, dissolvation term, etc. And you can also import some conservation of coevolution scores that are derived by the analysis of the sequences. And of course, you can combine these things to have a consensus method. So current scoring functions cannot accurately predict binding affinity. They're really struggling, and you may have very good now machine learning methods with very high performance, but it's not clear how they can really generalize to completely new data sets. Okay, so that's a more detailed representation of the Hex scoring function with this idea of interior skin and exterior skin. So you have these different terms, each corresponding to the overlap between exterior and interior of one, exterior and interior of the other one, and you have some penalty for the clashes. So what you expect is what you see on the right, where you have the exterior one touching interior of the other. Okay, so then if we go back to the original question, there was this idea of the arrangement of the proteins with respect to each other when we know they interact, but there was also the question of, can we determine whether two proteins interact? So for this, we can do a large-scale experiment where we take a set of proteins and we just dock them, all of them, onto each other, right? So from this, the goal is then to identify the true partners in this set, or the partners that we know, because of course there are partners we just don't know, we just can't say they don't interact. And so we can combine, how do we do this? So we compute we generate docking conformations for all these possible pairs. Then we also use the docking energy to filter them and to rank them. And then we're going to try to see whether we're going to identify the conformations that give you a good agreement with binding sites that we know or that we predict. So for example, if we have the experimental binding sites, we will compare the interface we have in the docked conformation with this binding sites, and we pick up the conformation that has the best agreement with these binding sites. Or if we don't have this knowledge, we may take binding sites that are predicted. So we have several methods that can predict binding sites. And so the idea is that then you can compute an interaction index for each pair using this best conformation. Okay, so you can just fill a matrix where you will have for each pair an interaction index. You can normalize these metrics by accounting for the global social behavior of the proteins. So let's say if a protein has very low interaction indices with all the other proteins, so it doesn't like to interact in general with anybody, then you will just put a bit up its value, meaning that if a value is just above the mean, you could consider it's very significant for finding its partner. And on the opposite, if you have very sociable proteins, you will just, by the normalization, you will reduce its interaction indices. So you can't put everybody equal. And this way you can identify the partners. So this is the description of the interface and the structures you can have, and find the overlap. I'm not going to go into too much details. But so we did this on a set of a few hundreds of proteins, and we could retrieve the true annotated partners in the set with a very high AUC, which is 0.81. So doing these things, you were able to identify the partners using docking algorithm and trying to answer the question 1. Okay, so with this, I think I finish, and I see you soon.